The overall goal of this experiment is to generate induced pluripotent stem cells, or iPSCs, from melanoma-infiltrating lymphocytes.
This method can help answer key questions in the field of tumor immunology, especially for cancer immunotherapy. The main advantage of this technique is that the ability to generate human iPS cells from tumor-infiltrating lymphocytes could provide unlimited numbers of less differentiated tumor-specific T cells for immunotherapy. This technique may have applications for the treatment of metastatic melanoma, as currently the poor survival of T cells in the clinic has been the major limitation of adoptive T cell therapy. This method may also be useful in the investigation of the T cell receptor repertoire in the tumor microenvironment. Demonstrating the procedure will be Dr. Kumiko Iwabuchi, a postdoctoral fellow from my laboratory.
After obtaining the tumor material, select 5 to 20 grams of tumor specimens and use scissors to dissect the normal, solid, and firm tissue from the fragile and/or bloody necrotic areas of the tumor samples. Mince the dissected specimens as finely as possible. Then, use a dissociator and a human tumor dissociation kit to disperse the minced tumor pieces into a single-cell suspension according to the manufacturer's instructions. At the end of the dissociation, filter the cells through a 70-micron strainer into a 50 milliliter tube.